There's still sediment on my skids.
Okay.
Alright, try that, Aaron.
Looks like this might
Coming
be
in.
one of the sponges that we imaged earlier, type of...
Got a jellyfish in Atalanticam currently.
Very
Yep,
smoky.
Just hanging out.
Yeah, come wide.
Come wide.
Yeah. Well, that sediment's just going to follow it in.
Yeah, Kahina, it might have been that the way we were seeing that the top of that white blob, that might have been an anemone instead of a sponge. That's a good point. We should take a look back at the imagery.
Oh, that's up quite high, isn't it?
What?
That jelly.
Yeah. Oh, I was just pointing it out in my camera. Not not suggesting you go find it.
Oh no, worth going to find it.
Todd's checking the winch.
Just give me skids a bit of a shake.
Yeah. Shake, shake, shake. Oh, a bunch of it just came off. Oh, a bunch of it just billowed off.
Yeah.
What? He's shaking the ROV around to try to get the
Just get the sediment out of the skids.
out of the skids. Yeah.
Might have some nodules in there when we come up.
Bring me back a fish, Dave.
One of those things you wanted to look at, Amanda?
Yeah, let's take a look at it.
Yeah, that'd be great. Thanks.
Right.
Brown, round, nondescript object.
See how we do here.
Unknown.
Okay, mate. Coming right. Whoa, 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 whoa.
Oh, we got something back there. Sorry. Oh,
Yeah, it looks like an isopod crawling along the surface of the nodules, but we're getting a closer look at this brown object here. Not totally sure if we're looking at a dead sponge or maybe a Foramanifera, a xenophyophore. If it is a xenophyophore, it's these tests are, they call them agglutinating tests. Basically, they grow and glue together organisms and sediment. The organism is a unicellular organism, so one cell, but it makes these huge
happy with that?
tests. Yep. Thanks so much, team. Not sure if that's what we're looking at, but great zoom in for further confirmation.
So good having that tilt back.
Are you muted? Sorry.
Yeah, it's so nice to
I
just
can
be
hear you,
able
but
to...
I can't hear you.
I haven't seen many of those isopods crawling on the nodules. So
No, that was a good
neat.
observation. Yeah, great to see which ones are found where. And Tina, one of our shoreside scientists, Tina Moldostova, had identified the isopod that was on the stalk, the old sponge stalk. That one looked like it might have been in the family Arcturidae. Something for us to look into later, get a good site reference. But there are a variety of different isopods that we've been encountering. Small crustaceans. Amphipods and isopods are related to each other. Distantly related to the shrimp that we've also been seeing.
How do you tell an amphipod from an isopod?
So the isopods generally are what we call dorso-ventrally flattened, so from your back to your front,
Go look for
squished.
tubes in
Okay.
this.
And then they have sort of similar appendages or legs throughout. Whereas the amphipods, again, these are general descriptions, what we're looking for. Amphipods that are more side-to-side flattened. So if you were to squish your arms together this way.
Oh, I see.
Yeah, laterally flattened, laterally compressed. And then their front legs and their back legs are distinct.
Okay.
So some things to be on the lookout for.
Awesome. I have to go for 2 interactions
Oh
in a
my
row.
goodness. Well,
But I'll be
enjoy.
back.
Hope they have some fun questions for you.
Let me know what I've missed when I'm back.
Oh yeah, for sure. Another one of those brown blobs going across the screen, unknown what it is. Looks like we're coming upon another urchin. I don't know if we can take a closer look at this little white spot speck.
Yeah, got it.
Thank you.
Come on, Gastropod.
Okay, here you go.
Zooming in.
Ah. Ooh.
Okay, you can mark a doubler in if you want.
Doubler. Yay!
Gastropod. If I can get it
And we
focused.
have a win. So this is similar to the gastropod that we were imaging earlier. Gastropods are snails. You can see the foot extended from the opening of the shell attached to the nodule surface. And it's feeding along that nodule, scraping along the way using its radula. Beautiful coil shape, very distinctive and hopefully diagnostic for our taxonomist at home. That's as close as we can get, yeah?
Okay.
Okay.
On the move.
Okay. Thank you. Nice spot. Beautiful images. These gastropods and other deposit feeders have a lot of cleaning up to do. There's a lot of dusting or snow powder, marine snow powder all over the nodules to eat.
They won't go hungry, that's for certain.
That's for sure.
It looked like its little squiggly trail was behind it.
Yeah, I know. Yeah, that was a good observation. Let's see, more crinoids as we move over the seafloor. Stalked crinoids are sea lilies, and more urchin tests. And a lot of these white dots could be those gastropods, or they might be the bivalves or clams that we were imaging too. Obviously we can't If we had all the time in the world, we would image and zoom in on every single white dot.
Like that one coming up on the left.
Or the one in the middle, or the one on the right.
They're everywhere.
I haven't actually been told how far we're supposed to go on this watch, so actually we could do a little more slowing down. Jacob, were you given specific homework as a navigator, making sure we got to a certain waypoint by the end of this watch?
Not really, because we kind of calculated out beforehand. We'll be like, okay, if we go this fast, and then as we get closer, we might like bump up speed. But in the beginning of the dive, it's pretty relaxed.
Well, if we can take a look at another one of these white dots, that would be great.
What is vessel speed at the moment?
It's 0.2, isn't it?
Are we at 0.2? Jacob.
Right now, we're going 0.2.
Yeah, no, we can slow down if they want. Works for us.
Yeah, so I think we're going to keep it at that. We can bump it up to 0.3 if we need later.
I
Oh,
see.
you got a squiggly
worm
thing in the middle of the...
Yeah. This
What I'm
looks like...
saying is if we slow down to 0.1, it gives us more time on this sort of thing.
That's right.
Yeah, it gives you more time with these. But I think if we did 0.1, it might be too slow.
Okay, go ahead, Aaron. You're going to have to get through that, I'm afraid.
Yeah, I'm leaning towards... It looks like a Pteropod test. So again, the different type of gastropod, they live in the water column, but their hard parts remain and settle out, sink out to the seafloor.
Okay.
Thanks, pilot, front
Coming
row.
on.
That's great.
Coming wide.
Yep. Top.
Oh.
But we do get plenty of homework before the dive, I'd say.
Ooh, big tooth.
Oh yeah, that's a big one.
Is that a tooth on the right or no?
Yep.
It looks kind of toothy.
Looks like it.
It looked kind of toothy.
Oh, I see it.
I think that was a holothurian.
It looked like it, yeah. Oh, I see cucumber.
Another tooth there on the right, looks like. Yep.
Can we take a closer look at this, pilot? It's a little stalked organism coming up. It's right here.
ahead, Aaron. I'll come round into it in a sec.
I wonder if that's a tunicate. That might be something different. And then a little carnivorous sponge in front of it. So the
There you go.
sort of organism in the background.
Go up to that.
I can't tell for sure,
Okay, you're starting to get behind.
Yeah.
but it may be a type of
Come
tunicate.
on.
Looks
Gotta
like
go,
there's some
guys.
animals attached to it as well. And then yeah, Cladorhizidae sponge in the foreground.
Swing starboard.
Yep.
Thank you. All these mysteries. Every time we zoom in on something different.
There you are.
So Jacob, is the feature that we're diving in right now kind of like a trough or a depression? When I'm looking at the zoomed out image of the map, it looks like kind of a bowl. Uh-huh. Yep.
Maybe on SPL.
Oh, I'm not on SPL.
There you go.
But yeah, so there's not a whole lot of elevation change, so it can be kind of deceptive when you look at the map here. I think these are 10-meter isobaths. But yeah, it's still all abyssal plain.
Yeah, just even small, modest elevation changes can make a big difference in terms of collection of organic material as well as flow and bottom environment. So some of these modest trough-like features can play a role in the composition of the animals that we're seeing.
Well, I'm excited
there
to
might-
see it.
yeah.
I know around like the waypoint 3 to 5, we should be kind of in that trough a little bit more.
Yeah.
Top left, can we take a look at that?
Yes, please. Looks a little more spiky.
I was about to say, I can see its feet from that far.
Yeah. It's all stretched out, going for a walk. Having some breakfast.
Does it have multiple mouths, or are those just to
It has a
stick on?
one mouth or oral area and one anus or behind. And then all of those extended pieces, appendages, are tube feet, modified tube feet that enable it to creep along the sediment surface or seafloor and also feed. So there's a lot of parts to these animals to help optimize their feeding and traversing of the seafloor.
Oh, okay.
I think your mic is a little far from your mouth, Dave. Oh, yeah.
Nice.
Beautiful. Thank you.
Lovely shot.
Coming wide.
But you don't have your fourth hand engaged, or?
Sorry about that. I was off SPL.
Yep. I think that's what she said.
Oh. All right.
Just trying to look up to see if we had possibly a better ID on that sea cucumber type of holothurian with really fat tube feet. And it looks a lot like the pennyagony that we've seen before in the abyss. That's my best guess.
It doesn't look too far off.
Yeah. The image that we have is a slightly different posture, but at least the appendages look similar.
Add that into the comments.
Thank you. Coming across some more sea lilies or crinoids. Again, scattered white spots on the seafloor could be a number of different types of organisms or remnants of animals that get laid down on the seafloor. Oh, can we take a zoom on this pilot?
Yep.
Let's see if it's something different.
Do we know how old crinoids are?
I don't, actually. We have a...
As an
Look it up.
individual or a
Yeah, I'll have
species?
to look it up.
As a species.
Oh, it's very
26.
old.
26.
Okay, mate.
I'm zooming in on this glass sponge.
With an amphipod, sorry, not amphipod, isopod at the bottom? Where is that one? No, it's an isopod.
Oh, looks like it. The glass sponge might be Hyalonema, sort of a tulip shape to it. You can see the pores, pretty obvious openings, and the osculum, the larger opening at the tippy top. And then the
Might
isopod
start to
hanging out on the
sediment
stalk.
up a wee bit there.
Okay,
Come on.
that was good.
Yeah, please.
Nice, thank you.
Go on.
Ta.
Someone mentioned Elassipodida.
Oh, for the holothurian. Okay.
Mid-Paleozoic era. What is that? Let's see.
So the Elassipodida is basically the order
Oh, hang on. my
of
thrusters have just quit.
holothurians. The Holothuroidea is a class, and then the next level down would be order
What you need?
Thrusters have just
Elassipodida.
stopped on
We're
me.
still
Okay.
looking at trying to
I'll
figure
note it
out
down.
what the genus is.
Looks like another glass sponge too. Yeah, he took off.
Pennyagony is a good, reasonable guess. It's within the family Elapidiidae, which is in the order Elassipodida. But we shall see.
To be
Gather
determined
our
later.
sea cucumber experts and have them ID all of our beautiful photographs, including the headless chicken, which
I'd
we
like-
haven't seen on this dive yet.
I'd like to point out I was smart enough to actually take a picture of the timestamp too, so I can find it a lot faster.
Oh, that's smart. Appreciate you gathering all those images. That's going to help us out a lot.
Absolutely. Trying to ID something is like 2/3 of the fun.
So the earliest crinoids, evidence of crinoids we have found that would match the current evolution of them are 480 million years old.
Wow.
Kinda young.
Living fossils, I think is
Yeah.
what they've been referred to. Yeah, that'd be great.
Yep.
Crinoids are living fossils. They've been around for a long time.
You got some floating past on the left too.
Yeah.
Yeah, a little larval house. Okay, Aaron.
Looks like a salp house, yeah, Larvacean house. Oh, neat. Zooming in on a sponge, glass sponge. It's kind of got like a cup shape to it.
Sometimes these sponges really look like mushrooms.
Yeah.
What
I don't know what those little white dots are.
Yeah.
Yeah, those white specks are interesting.
I don't know if they're eggs of somebody else.
Rice.
Rice. Really small grains of rice.
Sponges are known to be homes to other animals and can be a type of nursery. So finding eggs on sponges isn't too surprising, but I'm not sure if that's what we're looking at.
Okay, we can come
Thank
wide
you.
again.
Great shot.
Looks like a spoon.
Yeah.
Cheers.
It was a little broken, a little bent.
Actually, one of my sunflowers did that. It broke and then started growing like from the horizontally broken stem.
Oh, there we go. I think Sandy had turned the screen to nightlight mode, so it was very warm in color.
Oh no.
That's funny.
Was it hard to see?
It wasn't hard to see, but I'm sitting here, I was like, I've turned it down quite a bit. I
Oh.
feel like I should be back to normal. I'm looking at this image, I'm like, that is very orange still.
Oh no.
Something right here. Was mushroom things, or is it just sediment?
Okay, Aaron. Zoom on that bit.
It's down there to the
Well,
Yeah,
let's
right
look,
corner of the
let's look
screen.
at the between the lasers first. Yeah, the white thing
Okay.
that you're going in on. That'd be great. Thanks.
Hold that a sec.
Looks like
So it's
a
a barnacle
barnacle. Yep.
hanging out on the nodule surface.
There you go.
So none of these little white objects seems to be the same thing.
Yeah.
No
Pan
one
down a
size
bit.
fits all here.
There's something else here on the bottom right.
Need to be real quick. Where is it?
Bottom right, right there.
Tell me where.
So you see that kind of like T-shaped thing?
No.
Yeah.
Right corner.
Close to the top
Sort of
right corner.
starboard side.
It's closer to the bottom right corner.
Oh, bottom right.
Yeah.
Yeah,
just
it's
getting
going to mess
sediment
us up a
washed in.
little. Yeah, sediment's blowing that way.
See those? Just off the bottom of the screen.
Tilt your camera down, Dave.
Looks like kelp or something.
There you go.
Oh.
And then as soon as you get it, I would move on.
Yeah.
Dead sponge.
Yeah, I think we're good.
Sure?
Yeah,
All right.
thank you.
Interesting.
There's another white object on the lower right. Unsure what that was,
All
but
right.
take a look. Wondering what the type of barnacle it was. We've seen
And I'm looking down at you, so you're going
those
to have
found
to
in
go
the Scalpellidae family.
Boogie, boogie, boogie.
I don't know how to distinguish the different barnacles from one another, so that's a new learning
Ooh,
opportunity for me.
what are...
Looks like another brisingid sea star with its arm waving to us, saying hello,
Hey,
little
buddy.
Herc, hello.
Could see us coming from ages away with the lights.
I'm sure.
Yeah.
Slowly raised an arm.
That's
It's not waving, it's trying to block the light.
How are your interactions?
They both didn't show up,
Oh, goodness.
but there's one more.
Okay.
Hoping for better luck this
Yeah,
time.
yeah. Lots of things must be going on at home.
Right.
Well, we're glad to have you.
Wow. I'm happy to be here. Yeah, I
So
remember
good
my
interactions.
first morning I was doing interactions. The very first 2 interactions I had planned didn't show up.
Oh, no.
I was like, am I doing something wrong?
Oh.
But it happens sometimes. It hasn't happened since, so.
Yeah, you guys have been super busy. So glad that there's a lot of interest.
There's a ton of interest.
Yeah.
Doing great. Yeah. Hasta luego.
I think I actually just saw a
I'm ready.
bryozoa as we were going over it.
Somebody mentioned in the chat that they're being cut off from the stream, that the video transmission is down, and they hope all is well on the ship. All is well. They'll be back any minute now. All right, I'm gonna go for an interaction now. Hopefully, hopefully they show
Round
up.
3.
Hopefully the video will pick back up again. I'm checking in with my shoreside scientists to see if they're still able to see what we're seeing.
Just to the left of the lasers. You mind if we, or next to the lasers, mind if we stop and take a look at that? The white spot to the right of the lasers. Perfect.
Beautiful. We're checking out as many of these white spots as we can. Every time we zoom in, it seems like we find something different. Interesting.
There you go.
Did they? Okay.
Hang on.
Marking it down.
Yeah, we're going to move on.
Yep, that's all good.
Okay, looks like at least some of our shoreside scientists are able to see the video now. Maybe it was temporarily down. I don't know. Those things
Find
happen
another white
for
one.
sure.
It's real quick as well. It just suddenly...
Oh, it just sends you right back down into the seafloor. That one's funny too, because we're buoyant, so it's like actively.
Yeah. There we
Great.
go. Let's try this one here.
Oh.
Yep.
Yep.
Just doesn't want to pick up. Come on. There you go. Okay, Aaron.
Zooming in.
Whoop.
Looks like we're looking at a pteropod test, but on the left-hand side of the screen, we can see a foram that's attached to the nodule as well. So it's a good zoom to give perspective. Thanks, pilot.
Kick a little.
We're good on that.
Coming wide.
Still need to come a little bit further round.
That sort of brown plate-like structure that we saw growing on the side of the nodule is a type of Foramanifera, a unicellular organism. And the type is a xenophyophore. They grow in different shapes and sizes. Some of them are flat plates or like fans. Others grow into sort of round tests that almost look like sponges. Oh, here we go. Right there.
Yes. Yeah.
Do it.
Sorry.
Getting really excited.
This looks like, what do you think, Rye?
It's the rib cage.
We're getting followed by that cloud.
We saw this earlier,
I
yeah.
was going to say it's...
This is my new favorite thing that I don't know what it's called.
Oh.
It's a black coral, whatever it is.
Yep.
Okay, let me come around.
It is a
into
black
the wind.
coral.
Just hold that.
And Tino is describing that they kind of curve,
Okay,
the
mate,
branches
you're good.
curves.
All right, you gotta get the shot and move on.
And I'll get a closer look at the pinnules on each of the branches to see
It's got a
how
thing
they're
in it.
oriented.
Got an associate in there too.
Yeah, it's a beautiful, looks like an isopod associate on the branches.
So another
And-
Antipatherian.
Yep, definitely
Super
Antipatherian,
Um,
black
what was
coral.
that?
Yeah, I gotta go. We're
resembles
underneath
a-
Coming wide.
Antipathes.
Antipathes. That's what
Yep.
it was. I was like, it looks like the one we saw earlier, and I was trying to think of what the scientific name was.
Well, you got it. Antipathes.
I know, but I was going for the Antipathes.
All right, is it responding?
Because you were talking about the two different types and how
Okay.
they were,
You got it.
how they looked different.
Yeah.
I was trying to figure out.
So it's a good thing for me to remember since I'm learning down here too, like the
Yeah, it's okay. Coming in.
ones that the type of black coral that we've seen on this dive so far have kind of a coiled shape to them or rounded nature to the branches. How did Tina describe it?
Said
More
it's kind
like
of
a
basket shaped.
Basket shaped.
That's
right.
kind of cupped a little bit upwards.
That's right. Neat. Abyssopathes, which is pretty well named since we're in the abyss. And pathes is a part of the root of antipatherian. The broader group of black corals, as they're so named. It's the order of corals, Antipatheria. Is that your favorite, Rye? The ones that...
I think so.
Okay.
They looked very similar.
Yeah. Looks like another sea cucumber.
Couple of
on the lower
holothurians.
left.
2 holothurians, yeah.
More white dots.
It's like the pink one we passed kind of looked like the headless chicken that we were talking about.
Oh, headless chicken. Wait, wait. Oh, okay. I was looking at the windsock variety of black coral, and it definitely has more of a stalk or stem that's attached, and the branches of the coral are up above the seafloor. So that's distinct, easier to identify if you can tell how it's attached.
And then I believe Steve also mentioned if it looks like it's wind blown, it's usually also Antipathes.
You want me to zoom?
2 for 1.
If I remember correctly.
Wind blown.
Wind blown.
So you want to check these out here?
Yeah, that'd be great, pilot, thanks. I was settling in for a zoom on these white objects on the seafloor.
Okay.
Keep on getting surprised by what we're able to visualize down here. Oh, very cool.
Oh,
What do you guys think these are?
Polychaetes.
2 polychaetes.
Yes. Polychaeta have many parapodia. These are 2 types of annelid polychaetes. You can see their antennae kind of sticking out. I'm looking for scales to see if they are scale worms. I'm not sure if you can focus in more on one of them.
Sure can.
We'll go for that bottom one, stand by. Oh.
Nice. Yeah, I'm definitely seeing scales on the scale worm. These sort of flappy bits that you can see kind of blowing in the breeze. Distinctive characteristic of this type of polychaete.
I would think about
Neat.
getting a move on.
That was great. Great image. Thanks, guys. Thanks, gang. 2 for 2. So if it's kind of elongate, maybe it's a polychaete or a holothurian.
Swing starboard.
And if it's really small, it's probably a test.
Yep.
And if it's somewhere in between and still kind of rounded, you might get a snail.
Yeah.
There you are.
Or a barnacle.
Your heading's off now, eh?
No. Heading's good.
Oh, I mean on
Oh, on,
nav,
on
yeah.
the nav, yeah.
Yeah, it was good for a while,
Yeah.
but now it's back to...
This has been a really productive dive. I think I have felt like every time we zoom in on the seafloor, we see something a little different. We've seen some teeny tiny gastropods and polychaetes, a bivalve. I think that might have been the first one that I've seen for the
I think so.
expedition.
That was cool.
Yeah,
And a bivalve.
right.
In a pear tree.
I'm surprised you guys saw that. That was a good eye.
Thanks. We're aiming for white blobs,
Got a fish
Jacob.
over here on the right.
We're all about the white blobs.
The white blobs.
Zoom
what
in
is
on the
that?
white
What do you do
blobs.
for a
Ooh,
living?
fish.
Look at white blobs.
Yes.
Hello, fish.
Hello, fish. We've seen some corals and sponges and fish. Psh. Again, that
Nice little view of its head.
looks a lot like that. Maybe Leucicorus cusk eel. Definitely the next time we see a poop pile, we have some requests for taking a look at that too. So we're closing in on a cusk eel. Seems to be similar to what we've seen on the dive so far.
How do you tell a cusk eel and a halosaur apart?
Yeah, so the cusk eels, in my basic terms, the dorsal and the sort of anal fin, you can see that runs the length of the fish. You can see that sort of thin-like structure on both sides. And then on the halosaur, basically, that dorsal fin isn't prominent, but the anal fin runs the length of the fish and tapers out to a point.
Okay.
So, again, that's my non-fish expert eye towards trying to identify them, but for the most part, that's worked for halosaurs.
Yeah.
There's a lot of other ways to tell apart the different halosaurs, and it has a lot to do with scales on the face and shape of the face, other factors, but for a quick ID, that's the best I do.
Little crinoid or something on the right.
Yeah.
Another stalked crinoid.
That's fine.
Is the current still pushing you south?
Is there much of a current?
Just a little bit.
Okay. Yeah, I noticed the orientation, yeah.
What about that off to your port side there?
So a wee bit of a current. Oh, yeah. I don't know if you can see that over there. It went off the screen. That's okay.
Still quite a few urchins in the area too. Although fewer than when we started.
So I was saying that I guess next time we see kind of a poop pile or pile of fecal cast, we might want to take a closer look. There are a few different animals responsible for those, including acorn worms,
Okay.
but I haven't seen one in a while.
Did
Something
we
off on the right there.
did we ever
Holotherian.
identify what was in the tube? That was the
Oh,
swatch, right?
no, we have to look back through the video again. That sort of pop goes the weasel,
It haunts me.
little white thing.
It haunts
Might be
you.
a
Yeah.
sponge.
Sponge on the right. Looks like a different variety than we've seen on the dive so far.
Something's on it too,
Of a
something white.
glass sponge.
Or is it a holothurian? It's a holothurian with its head up.
Okay.
You want to check it out or not?
No, we
No.
can keep going. Thank you.
Oh.
All right. Aaron's the microscopic eyes. I trust him.
Yeah, I saw the gut and the dorsal stripe.
Nice.
Pretty dead giveaway.
2 urchins hanging out on the seafloor. Yeah, that's pretty amazing density of nodules, and then it seems like there's a lot of irregularity in the shape and size. It's a little different variation than what we saw on our first abyssal plain dive a couple days ago.
Yeah, they were nearly all the same, weren't they?
Yeah.
Guess the holothurians ain't eating very much up here.
Plenty of dust on the nodule surfaces. Fine powder of marine snow settling out. Deposit. It's interesting. I don't know if I'm seeing shadows as we pass over or something dark other than the nodules or something.
Something
Combination.
right ahead there? Or is that... No, it's just sediment pattern. Might be one of those like
Yeah,
bottle
it might
brush
be
type
worth taking a look at this.
things. The
Here.
brown fuzzy thing.
Yeah. Looks like a weird, unique pattern on the seafloor. Oh, there's a stalk of something down below
Right
too.
in there, yeah?
Yeah, like here.
Yeah.
Dusty dust.
Dusty dust.
Good?
Yep.
Taking a closer look, what looks like kind of disturbed sediment over here, see if we find any.
There is something to the left.
It's one of some castings there on the left side.
Looks like a poop pile.
But more left though, right?
Yeah.
Can we zoom in?
Left
What's in
and up
center for
slightly.
now?
Left to
Yep.
center?
Can we do the center for this
No.
moment?
Oh, for this
Yeah,
moment.
right there.
Thank you.
The little white thing.
Can telly on that.
Yep.
Looks like maybe another pteropod test, different shape. That's good.
Good.
Thank you. Maybe if
just
there's time to, I don't
zoom
see
out a
any,
wee bit,
there's
Aaron.
a pile of poop over here, but not sure I see the animal
Need to go
responsible.
up a bit and round to the left.
I think
there.
it's right there.
Oh, yeah, good eye. Going in?
Yeah, go on. We don't have long. Coming to
Tiny
you.
little glass sponge.
It is a tiny glass sponge. Definitely not responsible for making the poop,
No.
but but it is amazing to see these
Can you
size
double in quick?
size classes is variation in size of the
Okay, gotta
organisms
go.
we're encountering.
All right.
Beautiful,
Yeah.
great.
Starboard.
What's to the starboard?
Oh, just that's the direction he's got to scoot
Oh,
out.
okay. I thought maybe you had something
Once he loses sight,
exciting.
it's easy to get disoriented, so
Oh, gotcha.
I usually just try to keep track of what direction he was sitting before.
It's really interesting just the amount of things we find when we just keep zooming in.
I know.
You know, I don't think we've ever zoomed in and not seen something.
It's incredible, the amount of diversity at all scales. Totally agree, Jacob.
No, we were quite low. Okay. There we go.
I feel more confident that the round balls are urchins, but the specks, not so much. The white specks, not so much.
Does it stress you out a little bit that like no two have been the same?
It's oh, I don't know if we can look at this or if it's, yeah, anytime we see like a stalk, we might want to take a closer look at. We don't have to, if we can't get to that one, it's okay.
I think you'll probably just end up in
Sometimes it's
Thruster
hard to see it
wash
before we're over it. It's stress? No, I'm not stressed. It's more like I'm thinking about how we might deal with the annotating of the video, the collective community. So some things we'll be able to be confident that we're IDing pretty well, and then other organisms will not be able to identify without a close-up, and that is a natural kind of trade-off for the work you do with the video work.
In the annotations, do you use some sort of scoring system to identify kind of confidence level in?
Yeah, Aaron, that's a great question. Definitely, we use a confidence level, and then we also compare data collected from different annotators, so we can
like
see how well the data compare.
That kind of looked like a cookie cutter. A tiny little one that we just went over.
You can interrupt me. I'm just talking here.
I thought it was another teleos test, or Pteropod test until...
I'm just chatting with Aaron. You're allowed to tell me to stop talking. I'm looking out the window, but I have a hard time talking and telling people
using your eye.
Aaron has lots of questions.
Aaron's great.
Oh, there's a stalk thingy.
Oh yeah. Ugh.
Really,
Just like
really
right
tiny.
when we come right over
I
it.
know, they're hard to spot.
It's hard to spot.
Can run the iris a little bit hotter, might catch them sooner. All right, just going up to 80%. Looks like another bryozoan, or is that sponge?
Going
Over
off
on the
to
left.
starboard side a lot.
I think I saw us.
There's a stalk. Yeah, so I got to run the iris a little bit hotter there. So as long as you're okay, I can keep it below 80. All right.
This one kind of looks like carnivorous sponge.
Yeah, that's a carnivorous sponge, Cladorhizidae.
Oh.
Oh, actually, can we zoom in? Sorry, I might be incorrect.
Oh.
Since we're here, we might as well get a good look. Yeah, it looks like it might be
Going
a
in.
crinoid. Yeah.
That looks like one of those crinoids with the tendrils coming off.
Yeah.
Yep.
Nice.
Yep. Coming out.
I had those little mesostomes, parasitic polychaetes on the branches or arms as well. That's a great close-up.
Sponge on the right.
A sponge on the right. And
All right, get back into the center there.
so we're currently at 5,160 meters. Little Herc is diving here today in an area called Bissell Plain in the Cook Islands. The planned dive length is about 36 hours, deck to deck. We're on watch for another 30 minutes.
It's a stalked sponge,
So 4
it
to
looks
8.
like.
Exciting watch. We've had a lot of great animal encounters today.
And we found out that every single little white dot on the bottom of the seafloor is probably something different.
It's exciting.
Makes it a nice little challenge for
That's
us.
right.
We need to have a white dot bingo card.
Bingo card, that's a great idea.
Yeah, we
We're
should.
looking at a glass sponge on the
Deep
left.
sea bingo cards.
Side. Sure, Dave, why not?
Okay. Let
That'd
me
be
know when
great.
I can push in.
Looks like something's kind of dangling off of it as well. Not sure what that is. Looks like kind of tulip shaped, doesn't it? Oh, look at all those little critters hanging out on the stalk. Sort of-
Everyone's here for a party.
Yeah,
That looks
walking
so
along.
fun.
It's like a tightrope. Ooh, beautiful view of the top of the sponge.
Good with that?
Hexactinellid, I think it's a Hyalonema.
All right. Coming out.
And it has a really broad osculum or opening at the top.
With several mesostomid no. Were those still just isopods that were the tiny ones that were at the top?
Along the stalk?
Yeah, the tiny ones toward the top.
I
I saw the larger one at the bottom.
Oh, okay. Might be a few different species.
Something off to the left. Popping up.
Definitely seeing a mixture of substrate types here with nodules and sort of looks like crust. I'm not sure if we've taken a look, pilots, if you've taken a look-see at any kind of depressions we've left with the little Herc. See if there's like a thin layer of sediment underneath the nodules.
Oh yeah, that's a good call.
Uh-huh.
Yeah.
Yeah.
When I sat down.
Okay, so this is sort of a thin layer at that point. Okay, and that was, let's see, Jacob,
Stalk on the
are we
right.
starting to move into the trough area yet? Or not, we're not quite there.
Let me zoom out
Okay.
here.
Cloud of, yeah.
Yep.
We're definitely on the outer edge of it, but we're not inside.
Okay, thanks.
Do you want to zoom on this?
No, I think we're good.
Okay.
Thanks so much. But yeah, the substrate seems more varied than when we started the dive in terms of more kind of platey, crusty pieces of rock and then nodules. varied in shape, more irregularly shaped, whereas at the beginning it was more uniform.
Yeah, we are going like over a tiny mound.
Yeah, I
Kind of
wonder if this
rise.
might be like a deposit of some sort.
It it's not very big.
Yep, yep. Platey, crusty, moundy. Yeah.
Maybe like 200 by
Thingamabob.
200.
Thingamabob.
Yeah,
I love
yeah.
studying the
Usually a
thingamabobs.
thingamabob, though.
Thingamabob.
I like your way of saying it.
Thingamabob.
Thingy-mabob.
A thingamabob.
Is that a holothurian up ahead?
Yeah.
Nope. No, sorry. No, sorry. I put our depth as the winch tension, which is why I said nope.
No.
I was to myself. Oh. Yeah, yeah, I meant that too.
It was more flattened out than
Just
would expect
saying
for
nope on the
holothurian.
chat. So, are these, we're getting more different shapes of things and stuff.
That's right. The shapes of the hard substrate have changed.
Oh.
It's more irregular. That kind of heterogeneity plays a big role in the types of animals that we may be encountering, creating highs and lows on the seafloor at different scales. And Jacob was just mentioning they were kind of traversing an area that's sort of more more of a high rise than that sort of flatter field that we were encountering at the beginning of the dive. So it's a slight elevation change that can make a big difference.
Holothurian on them right there again.
Yeah, maybe can we zoom in on that, pilot? Thank you.
Good.
Ooh.
Yeah, so this is a type of, looks like glass sponge. And it's attached to a nodule, I think, to the lower left. And it's kind of tipped over. Looks like it might have fallen over and then started to grow upwards on the right. It's really got kind of lots of little and big pores. Quite varied shape.
Ooh.
Nice.
I like the shape of that one.
Thanks.
Perfect.
Yep, coming out.
Beautiful. Thanks so much, pilot. And front row. I think, yeah, just for the shoreside scientists, I think we're remarking that the layer of rock, the hard substrate here, seems to be pretty thin, and that there's sort of an under story of soft sediment below this thin layer of hard material. So it's loose. The hard substrate is varied in shape.
That's consistent with what we
size.
saw at the start of the dive. Yeah. Still what you're saying on the down camera?
Is that a bryozoan we're going over?
Ooh. Yeah, that might be a bryozoan. You're good.
Yep.
We'll mark it for.
I did not see what that stalk was that we just went over either.
Sorry?
There's like a long white curved stalk that we went over the
Oh,
top
okay.
of. Missed it.
Jacob, what's the track distance from start to finish for all the white waypoints? Is it 1,000 or yeah? How long is that again?
Could we stop and
Do you
take a
wanna know
look
the
at this
total distance
white spot
that
right
we're
here?
Sure.
traversing?
The distance for all the waypoints altogether?
Yeah, so it's 10 kilometers.
Okay.
Each
Similar
is just
to-
spaced by 1,000. Yeah.
Awesome. Thank you.
Going in.
It is, kind of looks like an, yep, it's just another urchin. Okay. Happy.
Coming wide.
It was smaller than the others, so I wasn't sure.
Oh, you're good.
I feel like we gotta look at an urchin like every once in a while, you know,
yeah,
just
to say hello.
yeah.
Yeah, they don't want to get them upset. No favoritism down here.
Just to check it's not a ping pong ball.
Golf ball. Looks like another sea cucumber on the lower left. An urchin. Center. Looking for more of those tiny stalks.
It looks like we'll make it to waypoint 2 before the end of our shift.
How many waypoints are there total?
10, I think.
Okay.
If we have time, can we stop at this little white spot as well? Center screen.
Right
Yep.
now.
Yeah.
Thank you.
Good.
You sound so sure.
Oh,
Ah, it's a
Sorry.
snail.
I was like, it is looking like a gastropod.
It was a snail, yep. Doot, doot. There we go.
Sure
That's a
is.
little snail.
There we go.
All right, we're going to push in with the doubler.
Gastropod. Its little foot
Ooh,
hanging
right on
out.
time.
Ooh, some iridescence there, too.
Lovely
Scary.
coiled shell. Very smooth in texture.
And he's waving.
And you get to see its antennae and its foot
Perfect.
crawling along the nodule surface. Beautiful.
Okay.
Thanks, front
Coming
row.
wide.
And so it goes. Another gastropod.
What did I miss?
Snail.
I
All
didn't
of
miss
it.
the snail. I
The
was here for
snail,
that
The
one.
whole
An
world.
urchin.
Were you here for the bivalve earlier?
Oh,
We found a bivalve.
a bivalve?
Yeah.
Yeah.
Shark.
So kind of like a clam that opens up, has 2 hinges.
Oh.
Found a bivalve.
I tuned in for a second when you guys saw a fish.
Hey, cusk
Fishy.
you.
Fish are always happy things.
If she saw Patrick, then I'll be upset because I missed it.
I'm glad I had Todd join me on that interaction.
Oh yeah, how'd it go?
It went well. Yeah. I learned a lot myself.
Oh, that's awesome.
It was one of those big college classrooms with like hundreds of students in it.
Oh wow.
Did anybody ask any questions?
Yeah,
Yes,
Good.
they did.
better.
Yeah, that's
They
better.
certainly asked questions that I wasn't able to respond, so I'm glad Todd was
cool.
there.
Is that a stalk there?
Yeah, looks like it. Stalk that stalk.
No, I think that's an urchin.
Yep.
Oh, but in front, closer to you, that's a stalk.
Just there. Yeah?
Sure.
Yep.
That one works too.
Yep.
Might just be one of the crinoids.
It does look like a crinoid.
Yeah, it's got a little bit of that yellowish tint to it, yellow-green.
Yeah, I think it's a crinoid.
Are we stopping or not?
No, I think we can keep wandering. But
Okay.
this level right here is great if you can fly like this.
Dragging my toes
Yeah.
through the mud.
Tilling the seafloor.
Just saying,
Yeah.
maybe not. Okay.
Lil Herc was here.
Yeah, little nodule
Oh,
collection
this
on the
looks
way.
like something's going on right there. There's
Yeah.
some action.
Jeez. Right, okay. I've got to spin
Sediment
right
excavation.
around that.
There's
Or we're
definitely
just gonna
some
get
sort of
wiped out.
trace.
Need the backup beeps.
Beep, 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 beep.
We have a Hey Nautilus, Scott Simon from MSI UCSB here with Franklin Elementary. Go Eagles! Our third graders want to know how deep you are, and we are at 5,164 meters deep.
I think Rye describes this just about perfectly as stuff.
Looks like there's some little holes in the sediment. Maybe burrows?
Oh,
Oh.
polychaete.
And a
Hello.
polychaete
Oh,
just
hello.
floating on in.
Yeah. Little action shot here.
They look soft. Are they soft?
They are squishy, yes.
So squishy.
So squishy.
I bet they'd tickle if they swam over you.
I think
See if
we're-
I can come
we're good,
around
pilot. You're
just a little to him. We may dust out though, because that was a really quick landing to get this.
Definitely
No,
saw
we gotta go.
You're good.
Coming
One
wide.
of the
Woo,
piles.
there he
Oh.
goes.
Hello.
So this is my
So
time
it's
to
my
run.
time to go
It's
too.
like TTFN.
Nice. One of the sediment pools that we were looking at just before we landed had some distinctive trace to it. Maybe there was some animal living in the middle of it, but within the sediment, so we couldn't see it.
We
Right,
made
you
it
just
to waypoint 2.
get on the end of that. There we go.
Two of how many?
10, I
10.
believe.
10. Awesome. Are we there yet?
I don't know if we can look at that on the left, pilot.
And just for that, okay, now you're going back to blue water.
Really quick. If I can get in. Coming down. So right
Oh,
Oh,
I
interesting.
think this is sponge.
Snaps in.
Okay, Aaron, go for it. Try and come
Interesting.
around a touch. There you go.
Looks like maybe dead sponge material accumulated here.
Okay. Coming up.
Yeah.
Detritus. Thank you.
R.I.P. Bobble polish. Okay, spin starboard.
Yeah. Truck out a little.
I'm very impressed. It's almost time for breakfast, and I haven't heard any discussion of food.
I have been strategically avoiding bringing up anything food related.
Learned our lesson yesterday.
Yay.
I think like within the 20-minute window of
I think
breakfast,
it's
you can talk
okay.
about breakfast.
Yeah, I thought we were like within that frame. Acceptable limit.
Acceptable breakfast talk limit.
Yeah.
All right.
Yeah, I'm not that hungry today. I'm not sure why.
Yep.
Although
Maybe
a crepe
shall come
sounds
down.
really
Read your
good.
mind.
Thank you.
If we talk about food enough, maybe I'll start being hungry.
I'm always
See
hungry.
what we get.
Good.
Too late.
Yeah, be quick. There's a little bit of dusting going on and the current's blowing right that way, so.
So should we focus on the holothurian
Looks
here?
like the holothurian is there. I thought I saw a stalk too, but this is good. Something's
Yeah,
up
I didn't
there
have
on the
time
base.
to get
Up
turned.
to the stock.
Okay, coming wide.
Looks
Yeah.
like something's at the base as
Go.
well. Neat. Thanks.
So that shows you just how soft it is underneath, actually.
Yeah.
Because that was a nice gentle touchdown, but stalk up there.
Yeah, if we can take a look at that.
A few stalks.
Yeah, a few stalks.
You get your choice.
Wait.
So someone's asking if we've seen any more of those worm tube things.
Still haven't found them. No more tubes.
Man,
We've been looking though.
yeah, we
have
have.
been
Find
looking.
him. I'll find him.
In fact, we've done a lot of stops to look really closely.
Let's see how that
Those were
works, Aaron.
unique, for sure.
Pushing
Ooh,
in.
this looks cool.
Oh, neat.
They're all lined up.
They're all lined up in a row.
Oh my goodness.
It's funny how they're evenly
That's
spaced.
so cute.
So those look like
Too
amphipods
close.
to me. 2 by 2, hanging out on the stalk.
Thinking, yeah, glass.
Yeah, glass sponge.
Yeah.
Oh, they all look like amphipods.
I'll clear in a sec.
Maybe
There you go.
Nice.
Oh, wow.
Hello.
that's lovely.
Oh my goodness.
Should
You're
be
like, what you looking
okay.
at?
Gotta go.
They're all looking right back.
I know.
Come port.
Lovely.
Come back
Yes.
to me.
Nice.
I miss you, Dave. Come back.
It's right next to you.
Oh,
Fish.
So
No,
Naves.
we gotta go.
and that is going to be listed as just a fish.
It's a fish. It was a fish.
It was a fish.
Naves is wondering, what would you like to find the most?
Oh,
What would you like to find the most?
a field of octopus.
Octopus.
Like an octopus nursery.
Yep.
That'd be
Yeah.
An octopus nursery would be incredible.
cool. Those are usually found, what, around thermal seeps?
Yep. Not something we're going to find on the abyssal plain.
Yeah.
I'd kind of like to find the Merchant Royal.
I mean, most of anything at all ever?
I think that would be pretty cool. Yeah. An octopus nursery?
We'd really
Think big,
push
though.
the
Think big.
doubler on the lens to its limit in an octopus nursery.
Should I think megalodon big?
Yeah,
All right.
megalodon is what I'm thinking.
Spend
Megalodon
some time
or
where you are there.
Yeah.
a crazy shipwreck.
Proof that they
All
still
of a
are
sudden.
around?
A crinoid fossil.
With a living
So
crinoid on it.
Pilot, are you
Yeah.
able to do a little bit of a drift test for me just to see how much of a current there is?
Can do.
And then Nav, can you see how far Pilot moves when he says go? We time it. We're trying to get an idea of current speed.
It's going to be really hard to give you an accurate read because
Plus or minus
whatever
is
we
fine.
do, we're going to get pulled by the tether,
Gotcha.
right? I'm dead stick now.
Okay.
And you're floating back to me.
Yeah. And you can see the current cutting across.
Mm-hmm.
Uh-huh.
I think the coolest thing would be finding a graveyard of whales. A whale fall graveyard.
Whale
So
fall would be
Current's
pretty cool
pushing
too.
you in what direction?
I think that's a lot more likely than my octopus nursery.
Someone said,
That's, again,
Sorry,
hard
can
to
you
tell. The current's actually going this way. If you look at the screen,
Yep. I'm just trying to
coming.
figure out which your Herc's
So-
orientation is.
Which would be somewhat south.
around about 3, 4, 5 coming from,
Okay.
right?
Yeah.
But I'm actually moving about 255. Sorry,
Okay.
225, just because of the umbilical and everything else.
Okay. So northwest to southeast?
Yeah.
Okay. That's helpful. Thank you. Sorry to interrupt the commentary. I just am trying to get a better sense of currents down here.
Yeah,
Not
no
at all.
worries.
we weren't having very substantial. I was going to say that someone commented they want to see a giraffe.
Oh,
So.
okay.
At
A
least it
giraffe
hadn't
fall?
descended into the realms of pineapple on pizza yet, like the
No.
last watch.
No talking
Pineapple?
about that.
What?
Someone said they like pineapple on pizza.
I'm starting to blacklist
Yeah.
topics.
Oh no.
I do like those
That's a, yeah.
personally. But
Dave,
we don't need to
you
go
done it
there.
now.
That's okay. That's okay.
Oh, sorry, I'll come down. Neat, neat, neat.
Oh, you're good.
I feel like visibility has gotten slightly worse,
No,
unless
we're a little
We're
bit high
pushing the
there.
high,
messing.
pushing the iris.
And
pushing the
the
iris.
iris
Okay.
is being pushed.
Yeah.
There's a casting. Do you still want to look at one of those, Amanda?
This would be worth checking out. A little more
Yep.
detail if possible.
What is this?
Doesn't look like a Herc was here trail.
Nope.
Yeah, the nodules appear to be sort of piled up on either side of this sand bed.
Like something larger might have burrowed through there.
It's quite the excavator.
Whoa, what?
There's
Yeah.
a pile of fecal material up ahead. Interesting.
Interesting.
But you
How
can
did
tell
that
that there
even-
is really a thin layer of nodules here and the sediments just below.
Like,
Right.
what the hell?
This is when all the animals come crawling across. Well, we can't see. Huh.
Oh, it's okay. Curious if like the current regime might be highly variable down here, which may, no, sorry, I'm not trying to explain what you just did, but I'm trying to describe the possibility for why we might see nodules piling up in certain places. So basically the community of folks who are tuning in, some of our scientists are saying it's interesting observation that we're seeing those piles and how that could be, what could be causing that. And animals can do that, but they also, the currents could also be a factor. So, but that depression that we just saw in the nodule field where the sediment was exposed, that looked really unique to me. Like something could have dipped in here and dug that out rather than necessarily a swift current directing that one spot. So I'm leaning towards animal rather than mineral.
I'd agree, actually. That's why I was kind of thinking, like, it kind of looked like something burrowed through there.
Yeah,
As if maybe resting on the seafloor for a little bit.
it's like it dug its way.
Yeah.
Oh, dune worm. Ooh. Oh, we got a little sea cucumber hanging out in the water column. Hello.
It's got little devil horns.
It's floating in the breeze, in the water breeze.
Rock
Looks
on.
so peaceful.
Looks like it has horns. Okay. It's interesting though that I'm thinking that the composition of the sediments must be kind of coarser grained material, because every time we've sat down and there has been some disturbance, the fine material settled out pretty quickly. So I think that means it's kind of on the heavier side, so more like sandier grain versus silt or clay. Yeah, if that's possible, that'd be great. Thanks, Dave.
Morning. All right, we're doing a shift in people. Have a great rest of the day.
Good morning. Good morning.
Poop.
Yeah, I wasn't sure if there was something towards the bottom, maybe. Oh, maybe it, look, it's come and go. Okay. That's good for me, pilot, thanks.
Go on, get.
Oh, we're having a watch change here. 4 to 8's heading out pretty soon. You guys are going to be in good hands with the 8 to 12. Lots of exciting critters to be seen. Good morning. All right, thanks team. Catch you later.
Test. Ooh, cool.
We're just kind of revving up the engine back here, but I'm not sure that was a complaint from Dave.
Oh, no, I know. I heard it. He was like, I miss it. I miss it. Can we take a look at, I don't know which one of these. That one. Yeah.
Oh, nice.
That looks right. No worries with me. I'm still doing my watch change. Yeah, I'm okay with that. On audio, quad sat, did that, rec 3, scope, client, yep. Oh. Yeah, go ahead. Coming in. Oh, I lost my mind focus too. Where is this?
Okay. So stalked sponge with an amphipod. Now, what kind of sponge is that?
Yeah.
What? Oh.
Copy.
Kind of looks like that big one we saw on top of the seamount last night.
Copy.
All right, you're good. We've seen it. Just don't quite know what it is yet.
Yeah.
What kind of sponge was that?
I think it might be the same that we saw, but smaller the other day. You remember that big one?
Yeah, except it didn't have as much structure in the back side of it, but...
Why did I lose
This
that?
one? Or
This one, the one we saw, you know, like at the top of the seamount had a lot of cool structure. This one didn't, but I bet someone on the science chat, I haven't even opened that up.
What's that Pokemon?
Caulophacus. Okay.
Caulophacus, that
It was
the
the
same
one.
one,
Yeah.
Caulophacus.
Facus.
Oh, fakers.
It's in the science chat. If you look at that, you'll see the spelling.
Nice.
Several subgenera of Caulophacus because the morphological diversity.
Thank you,
High.
Steve.
Some debate about the validity of subgenera, though. Oh, that's cool. Oh, looks like there are some big holothurians somewhere around here.
Yeah.
All right, stop.
Okay. Good morning to everyone across the globe, or depending on where you are, good evening. Welcome to the EV Nautilus. It's a beautiful morning where we are, at least. Probably to the sea creatures, they can't tell because it's really dark down there. Did anyone come up with a question that, because of Adam's exhaustion?
Favorite fast food
Favorite
place?
fast food place? Oh no,
What
I
is
feel
your
like I'm going to be
go-to?
exposed here.
Expose him.
Yeah. Alright. Kia ora, good morning everyone. My name is Anthony Vavia. I'm a Senior Research Fellow at Te Punawai Marama, the Cook Island Centre for Research in the beautiful island of Rarotonga and a lecturer at the Auckland University of Technology in
It's a gummy
Aotearoa,
squirrel.
New Zealand. And we have a very cool holothurian that we're coming up to. And
Gummy squirrel.
my favorite fast food, real quick, is Dirty Bird, KFC.
Oh, really?
Ooh, look at the track.
I'm in for the wicked wings, man.
Wow,
Nice.
that's gorgeous.
Not as wicked as this
Trucking.
holothurian that's got its hand up, wanting us to get its shot.
Gummy squirrel.
Gummy. Or
I've
maybe
never
its
seen the
little
gummy squirrel
appendage
now.
might be too small for a gummy squirrel.
Honestly, I think the gummy squirrel has like a double, like ears almost. So I think this is something different. but-
Yeah. This looks like one we've seen already.
Oh, for sure.
Just come from the buffet, too.
Yeah.
Yeah.
Just came from its favorite fast food
Yes,
takeaway.
the seafloor.
Sorry. Okay, Ed, you're good to go.
Thanks.
Oh man, what a giant! Holy cow!
Like an
Nice.
inflated balloon, eh?
Wonder what that
Oh,
oh,
little
Look at those
feet.
little feet in the front.
That's so cute.
Psychopodes longicauda.
Oh,
Steve Oskovich.
we need like slow motion Godzilla
Can you-
sounds here.
Oh my gosh.
Nice.
Turn the lasers off for me, please. Turn the lasers off.
Sorry, yeah.
There we go.
Oh, with the
Oh,
cute little
is this
Hyalonema
liftoff?
in the background.
Yoga. That's the Sphinx.
It might be.
Good
She's
stretch.
about to
Excellent
He
form.
better not take a dump on my ROV.
That's a warrior 2 pose.
Oh my gosh, it's looking right at us.
Hey guys.
Wow, what up?
Wow.
Oh, that didn't work out how I planned, but
It's like.
Yeah, it's not us doing that either.
No, this
Oh.
is a
Oh,
Chris Farley.
oh, it does have a
Holothurian.
double
I
little.
think that might be a holothurian food coma.
Throwing it back.
wow.
All right.
That is a bit different.
It's going to be the
So
latest
cool.
dance craze.
So graceful until it bonked into the seafloor. Oh, on the down cam, got a good shot of it. Amazing. That's probably a highlight of sorts.
Yeah. I would like to know which
Stalk.
one it is.
Oh, Psychropodes longicauda.
Oh, I just wrote Psychopodes. Okay,
Jessica
I'm going for my
say.
live interaction.
Okay.
Ta-ta.
Okay.
That was quick.
Smactilla.
Dropped
Ooh,
a dirty bird,
nice
then he left.
sponge, or is maybe with an anemone predating it. Can we make right? Give a. Oh yeah. Ooh,
Okay, we're going to look at that.
Yay.
Let's see if I can. Oh, getting used to Dave's. Yeah, nice. All right, here, setting. Whoa, he's. Oh, I lost my verts for a sec there. And then they just crash into the seafloor. That's gonna be
Perfectly,
great.
though.
Yeah, okay,
Well
go
done.
for zoom, because that dust cloud's going to get us.
Yep. Yep.
Okay, that's a stalk with a predatory.
It's about to go
anemone.
away.
Yeah.
NEM.
Wow.
All right.
What?
It's really
he got
cool.
on, that's why.
Oh, that
Because he had
anemone
auto
is
depth on.
psyched. It's like, yeah.
Can you
Yeah.
turn the lasers off for a sec?
Yeah.
Thanks.
Get a good shot.
No, I
Alright, sorry
actually
about
like
that,
having turbine shots highlighted
Yeah, I
like,
gotta
myself.
show-
Yeah, no
Yeah.
kidding. That was like
Sorry, I just gotta get my Z bias and stuff.
deep sea Dukes of Hazzard there.
Super. I wonder if you had that on a lot.
Shrimp?
Yeah.
Sea spider?
Okay, go for zoom. We're
Holy
gonna float
smokes.
this.
Holy
Yeah.
smokes.
So we saw one of these at the very start of the dive.
Oh, that wasn't a sea spider.
Okay,
Maybe
I
it
gotta
was.
get up
Yep.
ahead, guys.
Yep.
Okay, I'm gonna get oriented and then we'll...
Zip.
Sorry, handover's always a little...
Sorry, Rai, can you put the lasers back?
Yes, lasers
Thanks.
coming on. And can everybody hear me all right?
Yes.
Yep.
Okay.
Loud and clear.
Okay, I'm just gonna do a check if I like my Z bias and everything, and then get pushed out ahead here. Okay,
You don't say Z bias?
I don't.
Does Josh say that bias?
No.
Okay.
Yeah, that was super odd. I don't think so. I do think I've got sediment on my treads right now, though.
A car wash?
Yeah.
Just
Hopefully
wag.
just zooming around. Well, sorry,
Yeah.
this is gonna be really disorienting, but
It's like,
yeah.
like when the dog shakes its head. Ruh, ruh, ruh.
Which I just don't want to keep on my...
While we're car washing, there's
Okay.
a question for ROV and maybe science.
What?
Does it make sense that the pilot has the sole control for lasers on and off? Couldn't that be a delegated thing?
Yeah, that the pilot doesn't really
Okay.
need them at all.
I'm happy.
Right.
Doesn't care if they're on or off.
Yeah.
I guess they're just...
So couldn't we in C-Log put a lasers on and off button or something, or whatever interface you're on most often back there?
The one thing about doing that is then it would make a log entry of lasers on.
Well, no, I
Checking a
mean
page
just
out of your
somewhere
book,
in the
I like
web
that.
GUI,
Yeah,
put a button.
I guess the other
I
concern
Not
wasn't
a
doing
log
that before.
entry.
No.
to keep it in the
it's way
front
better.
row is like, what if you have a
Yeah.
ground fault
Secure.
on the lasers?
Well, I'm
or
not saying remove it. I'm just saying have it.
add it.
Yeah, I'm
Yeah,
gonna
yeah,
try
yeah,
that one
yeah.
so I feel
Yeah.
a little more.
Yeah, I mean, I guess it could make a log entry.
No,
but
not a log entry. There's a button in there, and all it does.
no, but everything in the web GUI
Spread out here.
gets a log
That's
entry.
okay. We just have a...
Do you have another computer interface you look at?
There used
besides-
to be, for the Triclops, there was a little, what do you call those things? A little button.
Yeah.
Oh yeah.
array.
What about one of those?
Yeah. Boop, boop. I would like
Just
that.
one single one with a big laser button.
They make tiny ones. Stream Deck.
Yeah, Stream Deck. That's right.
I like the idea of having a controller with only one button.
Like a big red button.
Yeah.
It's like if you get in an
Lime
elevator
green button.
in a building with
Laser.
2
Green
floors,
button,
and
yeah.
they have a button for each floor instead of just having one that says
Yeah.
go.
What
Okay.
if they're...
Do you want to go for zoom on
Was-
this little white speck?
Yeah,
I'm
I'd
just
love to,
testing
love to.
some.
How about like one of the old style rotary phones, and if you pick up the
You can
receiver, then it goes off.
snap in
If you
more.
put it down,
Ed.
it goes on.
You want?
Yeah, that would be
I'm going for the little
interesting.
white speck.
But we'd have to use a red phone to make it
Red
look cool.
phone, yeah.
Why not lime green?
Maybe
You wanna
more of a
push
mint?
into that speck there, Ed? Go for zoom.
This?
Yeah.
Oh, you know what that is?
Is that a gastropod? Is that what they
No,
were
I
saying?
think it's... They said it's been something different every time. I think that's a little sponge.
Hmm.
Oh.
All right. Come wide. I'm happy with how much sediment that didn't blow up. That's nice. All right, on the move.
Moving along.
What is this watch most interested in seeing? The last watch was very interested in little white specks.
We're
not that into those, but that's okay.
Cool.
Yeah.
Okay.
Because the last watch seems to have thoroughly investigated them.
Okay.
And also they don't qualify as megafauna. So.
Oh, okay.
I
Good
kind
to
of
know.
forgot that we are actually in the process of doing introductions, and Tonga has suggested our favorite fast food place.
Oh, wow.
Are
we more interested in stocks?
More interested in stocks. We're stalkers back here, you know,
Cool.
into the stocks.
slightly interested in the urchins, but we don't want to feel too bad about
Yeah.
So I'm Adam Sewell. I'm a professor at University of Rhode Island, Graduate School of Oceanography. I'm into stalks. Live in Cape Cod, Massachusetts. And
Go for Zoom.
Coming in.
I'm sitting in this science row here on the 8 to 12 watch. My favorite fast food place.
And I'm gonna bring my heading around a little bit. There we go.
Caulophacus? Oh, I think it's the same one.
Caulophacus, yeah.
Does that have to be a chain?
No, in
Oh.
fact, I'm trying to avoid chains.
Do you want a closer look at this, or?
No, we know what it
Yeah,
is.
yeah.
Roger. Okay, come wide.
I'm going to go with a place called Big Tom's in Olympia, Washington. Not sure it exists anymore.
Big Tom.
Big Tom's, you get a Big Tom burger. There was a fish
nice.
burger Friday. There was a corn dog Tuesday when I was
Corn dog Tuesday.
it was 2 for $1, but this was back in 20 years ago. But it was priced right for high school Adam.
Nice.
There's a stalk.
A little one. Oh,
I don't
think.
I think
I think
that's
that's just some schmutz. Dead stalk.
We'll
Dead
never
stalk?
know. No, I don't know. Let's look. You never know.
Oh,
Okay.
maybe it's
Never know till you know.
a dead
go for
stalk with
Zoom.
hydroids on it.
That's your prediction.
Dead stalk.
Dead stalk. With the white speck
Nice.
behind.
Do you want any closer?
Nope.
or-
Nope.
Okay. Okay, gotta push out a little bit.
Urchin.
Got a little stalk here.
What I really enjoy when
I
Rye
went
is
past it a little
piloting
bit, sorry.
is that she talks us through everything she's kind of like thinking about, and it's actually really helpful.
Is
to
it?
me.
Okay,
Yeah.
it's not annoying.
That and her sound effects.
Yeah. I am looking
There's a
at this
crinoid.
little stalk here. It's almost invisible.
Crinoid.
Okay, go for zoom.
Oh. It's like we tell Rye that we're interested in stalks, and they're going to find every single stalk.
Yeah, okay, that one
Dead or
looks
alive.
Nothing.
Nothing.
Oh, let me get a tighter shot.
Not even an amphipod.
I'm coming up. That's pretty bleak. Usually they have at least something on there. Yeah, I have tuned to the stock station now, so. You want stocks? I got stocks. Not the kind that are financially
Yeah.
beneficial.
Rye's a day trader today.
Yeah.
Someone said, yeah, don't give laser control to the second row because they'd make a laser
you interested
light
in
show
that
for
one?
funsies.
May,
What's that?
do you want to see this one?
Oh,
Yes.
yeah, that looks like a coral, no?
Kind of looks like it, or
Yeah.
Let's check it out.